I'm a biologist, and so I will speak more about the biology of multiple myeloma. I am not a clinician, and I will try to summarize some data that have been reported about the role of AGF and its receptor in symmetry, multiple myeloma pathogenesis, and in drug resistance. We know that multiple myeloma is still an incurable disease, and despite a significant improvement in the treatment, intrinsic and acquired resistance of malignant B cells to pharmacological treatment still represent an obstacle for survival improvement. The complex interplay between multiple myeloma and accessory cells of the tumor microenvironment are critical in supporting differentiation and expansion of the malignant clone, either directly by cell-to-cell interaction or also indirectly through the release of soluble mediators such as cytokines. Among these mediators, we have focused on the role of hepatocyte growth factor, because during the last 15 years, it has been shown that hepatocyte growth factor is important and is involved in the multiple myeloma pathogenesis. And also we before have studied hepatocyte growth factor and CMET in another type of lymphoproliferative disease, in particular in chronic lymphocytic leukemia. So I will try to present an overview of the data that have been reported, and I will try to convince you that about hepatocyte growth factor and CMET, this axis is also important and can represent a target for the cure of multiple myeloma. So here there is the schematic representation of the CMET, that is the receptor of AGF, that is formed by an heterodimer that resulted from the cleavage of the 170 kD protein in a 50 kD alpha chain and the 140 kD beta chain. These two chains are linked from a disulfur binding. Upon AGF binding, CMET dimerize and result in a transphosphorylation of two tyrosine residues that are in the kinase domain, that are followed by autophosphorylation of two other tyrosine residues in the C-terminal region. The phosphorylation of Y1349, Y1356 creates a multi-substrate docking site that is necessary for the induction of downstream signaling. And you can see there is a plethora of signaling that can be engaged, such as the pathway of RAF, RAS, RAP, MEC, and MAP, as well as STAT3, STAT5, and P3K, AKG. And this pathway also drive distinct biological signals, distinct biological response, such as motility, proliferation, cell cycle progression, migration, invasion, and adhesion. It has been demonstrated. We know that AGF is produced by bone marrow stromal cells, but it has been demonstrated that in multiple myeloma patients, there is a higher levels of AGF in the sera, and these are higher in this patient than in normal controls. And the researchers try to understand if AGF could be also produced by malignant plasma cells. And in the past, they, Borsay et al., a collaborator, have shown that demonstrated that malignant plasma cells secrete AGF and contemporarily also express its receptor, the tyrosine kinase CMET. It is also interesting to note that the AGF production is increased when multiple myeloma cells are co-cultured together with bone marrow stromal cells, indicating therefore that these interactions stimulate the AGF release. In addition, it has been then demonstrated that the multiple myeloma plasma cells also express the AGF activator, thus suggesting that the AGF proliferation of malignant plasma cells may also involve an autocrine loop. AGF, as I have also said, is produced by bone marrow stromal cells in the tumor microenvironment, and AGF thus provides, produced by bone marrow stromal cell, provides an additional paracrine loop that stimulate and favors the growth. AGF that is produced within the tumor microenvironment induce three kinds of response, such as mediate three kinds of response, such as survival, angiogenesis, and immunosuppression. In fact, Deckerson have demonstrated that AGF has a strong proliferant apoptotic effect on multiple myeloma primary cells or in cell line. And then it has been also shown that the inhibitor of AGF, ARC-196, overcomes the anti-apoptotic support provided by the stromal NK cells on multiple myeloma cell lines when they are co-cultured together. AGF is also effective in angiogenesis. Angiogenesis, that is a constant hallmark of progression in multiple myeloma. It has been, in fact, demonstrated that endothelial cells from bone marrow multiple myeloma patients express significantly higher levels of AGF and c-Met as compared to healthy control. And in addition, the AGF c-Met pathway appears constitutively activating in endothelial cells from patients, and higher phosphorylation of c-Met has been detected in relapsed and refractory multiple myeloma patients. AGF also contributed to the regulation of the immunosuppressive state in multiple myeloma plasma cells as well in cells of the microenvironment. In fact, kynurenine is, the levels of kynurenine is very high in sera and in fluid from bone marrow multiple myeloma patient. And it has been also shown that the expression of IDO1 is higher in multiple myeloma cells. Since AGF induced IDO1, it was suggested that AGF may induce IDO1 on the multiple myeloma plasma cells. AGF is also involved in the bone disease in multiple myeloma. We know the multiple myeloma patient develop localized and generalized bone loss. And during the disease, when the disease progresses, the osteoblasts become fewer and less active. And so we know that IGF is stimulate osteoclastogenesis, but some researchers also identified AGF as a potential candidate that could mediate the inhibition of osteoblast differentiation. After this, it was established also a positive correlation between the AGF expression in bone marrow microenvironment, multiple myeloma patient, and the extent of bone disease. I would like to introduce here also a paper that has been recently published. In this paper, the authors have tried to understand why malignant plasma cells may induce osteolysis in one region of the bone, and there is no sign of bone destruction in other regions, despite the infiltration of subtotal infiltration of plasma cells. So the authors here have, through single cells RNA sequences, have compared the plasma cell that they obtained from bone marrow aspirates and plasma cells obtained from the region of osteolysis. And through this comparison, I like to show here that they have individuated the upregulation of genes that are associated with osteolytic lesions. And among these genes, there is also AGF, DKK1, AGF, TIMP1. As we can see here, there is the representation, the differential expression between osteolytic lesion and bone marrow. And we can see here that AGF, TIMP1, DKK1 are overexpressed in plasma cells that are derived from osteolytic lesion. It's also very interesting to see this picture in which there is the comparison and between plasma cells derived from bone marrow and osteolytic lesion, and we can see that AGF is also here expressed at higher levels in comparison with plasma cells from bone marrow. So at this point, we can ask if the inhibition of AGF CMET axis can be helpful in counteracting the drug resistance. Have been developed in the past many studies in vitro and preclinical studies that have showed encouraging results. And in fact, as we can see here in this paper from Moschetta and al. And collaborator, they evaluated the activity of the c-MET inhibitor SU11274 in multiple myeloma cell lines that are both sensitive and also resistant. And so you can see that when the cells were contemporarily treated with bortezomib and the anti-c-MET inhibitor, the c-MET inhibitor can revert to the resistance to bortezomib, to melphalan, and to doxorubicin. In agreement with this data are also the observation that the silencing of U266 cell line by sirna c-MET, these cells show a higher sensitivity to bortezomib and to doxorubicin. In another study, it has been also shown that another c-MET inhibitor, not ATP competitive, tivantidine, could reduce the proliferation of multiple myeloma cell lines, and tivantidine appear equally effective in inducing apoptosis in multiple myeloma cell lines resistant to melphalan, to dexamethasone, or to bortezomib, or to lenalidomide. Then, based on these encouraging results, some groups conducted some clinical trials. And so I show here two papers that are the report of these clinical trials. And in this paper, the authors have used the tivantidine in patients with treated patients with relapsed or relapsed and refractory myelomas whose disease had progressed after 1 to 4 prior therapies. And they have shown that only 4 of 11 evaluable patients, so the 36%, had stable disease as the best response, while the remainder show a disease progression. They didn't detect partial remission or complete remission. In the other paper, the authors show the result of the treatment with cabozantinib, that is inhibitor of multiple receptor tyrosine kinases implicated in tumor growth and neurogenesis. The primary targets of cabozantinib are the hepatocyte growth factor receptor protein, and also the vascular endothelial growth factor receptor 2 and RET. And so cabozantinib maintained a stable disease in 8 out of 12 patients, while they don't detect remission. In spite of these not very promising results, that however employed only the anti-c-MET as a single agent, the author suggested that it would be rational to consider the use of c-MET inhibitors in combination with other anti-myeloma therapeutics, since c-Met could be associated with drug resistance. I would like here to introduce other novel strategies that have been developed to target c-Met, such as the targeting Samirna, because it has been shown that in multiple myeloma plasma cells there is an imbalance in the expression of Samirna, and in particular it has been identified Samirna that are related to the expression of AGF-c-Met, and these Mirna appear downregulated. So it has been proposed to use some Mirna mimics, and also some different antibodies that may compete with the AGF-c-Met, such as MK4, or these antibodies that bind simultaneously with AGF and AGF, or also a monovalent anti-c-Met nanobody. This kind of antibody have been tested in vitro, showing to efficiently inhibit AGF-mediated survival, proliferation, and migration. In addition, would I also now to introduce this paper that has been recently published, because it has addressed the circular RNA, and in particular, here the authors have shown that this circular RNA, circuitic, is overexpressed in multiple myeloma cells in respect with healthy donors. And this circuitic is regulate one Mirna, that is MIR32455P, and in particular circuitic is overexpressed, but downmodulate this Mirna, that in turn upregulate AGF. So AGF may induce, again, proliferation, migration, or induce downmodulation of apoptosis. And so they propose this molecule that contribute to multiple myeloma progression. Here there is a schematic representation of what we may propose. So the combinatorial therapies that may target c-Met or AGF through different bioactive molecules, such as antibodies or inhibitors of the c-Met tyrosine kinases, or also they may target Mirna or utilize Sirna in combination with the classical therapies. So we propose that these combinatorial therapies may shed new lights in counteracting drug-resistant multiple myeloma. So finally, the findings of an abnormal activation of AGF-c-Met in malignant plasma cells, as well as the multiple myeloma microenvironment that appear in direct correlation with progressive disease, may provide support to the use of drugs that may target this pathway to break the crosstalk between myeloma and the accessory cell, and possibly to restore drug sensitivity. Since the result of a limited number of clinical trials do not seem too promising when anti-c-Met drugs were used as a single agent, we proposed the combination of these molecules with conventional multiple myeloma therapies that could be useful to power up the current ongoing treatments. And the availability of a reliable technique capable of evaluating the levels of AGF-c-Met activation and the better compression of the feature of patients who will benefit from c-Met inhibitor treatments, along with the development of next generation bioactive molecules, could speed up to devise novel strategy to overcome drug resistance in multiple myeloma. So thank to all of you, and in particular, I would like to thank Professor Paolo Giannoli that is here with me, and we work together, and he gave also support to this presentation. Thanks for your attention.